T cells are needed to control intracellular pathogens and to activate B cell responses to most antigens. Pathogens are accessible to antibodies only in the blood and the extracellular spaces. However, some bacterial pathogens and parasites and all viruses replicate inside cells where they cannot be detected by antibodies. The destruction of these invaders is a function of the T lymphocytes or T cells, which are responsible for the cell-mediated immune responses of adaptive immunity. Cell-mediated reactions depend on direct interactions between T lymphocytes and cells bearing the antigen that the T cells recognize. The actions of cytotoxic T cells are the most direct. These recognize any of the body's cells that are infected with viruses which replicate inside cells using the biosynthetic machinery of the cell itself. The replicating virus eventually kills the cell, releasing new virus particles. Antigens derived from the replicating virus are, however, displayed on the surface of infected cells, where they are recognized by cytotoxic T cells. These cells can then control the infection by killing the infected cell before viral replication is complete. Cytotoxic T cells typically express the molecule CD8 on their cell surfaces. Other T lymphocytes that activate the cells they recognize are marked by the expression of the cell surface molecule CD4 instead of CD8. Such T cells are often generically called helper T or TH cells, but this is a term that we will use for a specific subset of CD4 T cells. CD4 lymphocytes can be divided into two subsets which carry out different functions in defending the body, in particular from bacterial infections. The first subset of CD4 lymphocytes is important in the control of intracellular bacterial infections. Some bacteria grow only in the intracellular membrane-bounded vesicles of macrophages. Important examples are Mycobacterium tuberculosis and Mycobacterium leprae, the pathogens that cause tuberculosis and leprosy. Bacteria phagocytized by macrophages are usually destroyed in the lysosomes, which contain a variety of enzymes and antimicrobial substances. Intracellular bacteria survive because the vesicles they occupy do not fuse with the lysosome. These infections can be controlled by a subset of CD4 T cells, known as Th1 cells, which activate macrophages, inducing the fusion of their lysosomes with the vesicles containing the bacteria and at the same time stimulating other antibacterial mechanisms of the phagocyte. Th1 cells also release cytokines and chemokines that attract macrophages to the site of the infection. T cells destroy intracellular pathogens by killing infected cells and by activating macrophages, but they also have a central role in the destruction of extracellular pathogens by activating B cells. Most antigens require an accompanying signal from helper T cells before they can stimulate B cells to proliferate and differentiate into cells secreting antibody. The ability of T cells to activate B cells was discovered long before it was recognized that a functionally distinct class of T cells activates macrophages, and the term helper T cell was originally coined to describe T cells that activate B cells. Although the designation helper was later extended to T cells that activate macrophages. And that's T cells are needed to control intracellular pathogens and to activate B cell responses to most antigens. Have a great day.